Welcome to the video abstract of my poster with the title Microfluidic Synthesis of Liquid Crystalline Elastomer Actuators. In liquid crystalline elastomers, or short LCEs, the mesogens of a liquid crystal are attached to the polymer network of an elastomer. By destroying the liquid crystalline phase, for example by heating it above the phase transition temperature, the polymer backbone can relax from an anisotropic stretched conformation into an isotropic coil form, which leads to a macroscopic deformation of the whole sample, making this kind of material an interesting candidate for an application as actuators. As an alternative to conventional LCE fabrication methods, monodisperse and actuating micron-sized particles can be made via microfluidic techniques. In our setup, the monomer mixture first forms droplets at a capillary's tip and is polymerized farther downstream. Hereby, the microfluidic process fulfills two requirements at the same time. On one side, it induces an orientation of the liquid crystalline director in flow, which is necessary to make actuation of the particles possible. And on the other side, it produces a multitude of equally sized particles. Using this microfluidic setup, particles showing a strong and reversible actuation were obtained. And by introducing azobenzenes to the LCE system, we also succeeded in the preparation of photoresponsive particles, which show a full actuation of up to 24% in less than 10 seconds during irradiation with white light, as it can be seen in this video here. This deformation is reversible either by keeping the particle in the dark or by irradiation with red light. Recently, we focused on the synthesis of more complex particle morphologies. With the aid of a modified microfluidic device, we managed to prepare LCE microtubes by polymerizing elongated Korshell particles. These microtubes show a thickening of the tube's wall upon heating, which leads to release of an air bubble that is trapped inside. Currently, we make these tubes photoresponsive in order to get light-driven micropumps. This works by moving a cross-sectional tapering induced by light along the tube, which should cause perfusion of a liquid inside. First experiments prove the successful photoactuation of these micropipelines already. We are also working on the preparation of dual responsive Janus particles, which, beside the LCE site, have a poly-NIPAM site as well. Latter should shrink by heating the particle above the microgel's LCST, and the other side should show a contraction by irradiation with white light, so that a second actuation of the same particle can be achieved. If you want to learn more about this very interesting topic, I would be happy to welcome you at poster number 1.